In this video, we will discuss about anti-tumor efficacies of oncolytic herpes simplex virus. So, in the previous videos, we have understood that this oncolytic herpes simplex virus act on the tumor microenvironment, but there is no detailed studies available before this paper about the mechanism of this, how this oncolytic herpes simplex virus act on this TME of the cancers. So they have developed a model which is 1:10 mixture model that has one part of the KPC cells and 10 part of the pancreatic stellate cells. So here they are checking efficacies of this oHSV in these two different models, a KPC model which is the control model and 1:10 mixture model, which is their model that can replicate the TME similar to the in vivo based on their analysis that we have discussed in the previous video. Now they took this mice with this KPC model or 1:10 mixture model, and after 6 days they inject this oHSV at the 3 days interval of 3 times and then check the survivals. So when they use this oHSV in the 1:10 mixture model, they are able to see suppression in the tumor volume compared to the KPC model case. And in the KPC models also they see some suppressions compared to the control, but those are not significant. However, when they check the survival rate, we do not see any significant difference in the survival probability of survivals for any of the case. So indicating that even though the oHSV suppress the tumor volume in 1:10 mixture model, it does not finally help in this increased survival probability. And individual results are shown in this panel D, where we can see that for the control, that is KPC tumor with PBS and KPC tumor with oHSV, there is not much significant difference, but there is slightly difference. However, when they check with the 1:10 with PBS and 1:10 with oHSV, we can see a significant difference. That is also mentioned in this panel B. So this indicates that oHSV works better on this 1:10 mixture model compared to the KPC model. However, now they wanted to go more deep inside this to find out why this kind of observations they are able to see. So in the next experiment, which is similar to the experiment that they have shown in the panel A, the only difference is that after 3 days after this 3 days after the final injection of this oHSV, they took the tumors and analyzed them in the flow cytometry. What they are able to find out that in both the case, that is in the KPC case and in the 1:10 mixture case, this myofibroblast cancers, myofibroblast CAFs goes down, and myofibroblast has the immunosuppressive properties. They goes down in the KPC model, but they goes down furthermore in the 1:10 mixture model. So this could be the reason that maybe the oHSV is more active in this 1:10 mixture model. And when they check it for the induced CAF, although it shows a decrease in the KPC and increase in the 1:10 mixture, but those decrease and increase are non-significant. So they do not put much attention on this induced CAF. And when they next they check the antigen presenting CAF, and this antigen presenting CAF seems to be upregulated, which is observed in both the KPC and 1:10 mixture model. With KPC models and with this 1:10 mixture model, the increase is more significant compared to the increase is more compared to the KPC model. So this indicates that this also helps us to know why this 1:10 mixture model shows high response against oHSV, because it has more antigen presenting CAF, and this antigen presenting CAF has the ability to be detected by the immune cells better. So moving forward, they checked different markers of the antigen presenting CAF, that is MHC-I, MHC-II and CD86. All these three are upregulated, and in all these three cases, the upregulation is in the 1:10 mixture model is significant, is much more higher compared to the increase that they have seen in this KPC model. In the case of these MHC-I and CD86, they do not see much change in the KPC model. Only antigen that they have seen the significant changes is MHC-II. However, in the 1:10 mixture model, they see a change in all the three cases. And when they check it for the other markers, other antigen presenting marker, that is CD80 and CD40, they do not see significant changes in all of the cases, in both the cases. So this indicates that it's this MHC-I, MHC-II and CD86 that are enhanced in this when the one-to-one infection model is treated with oHSV, and that helps in this enhanced tumor suppressions, whereas CD80 and CD40 does not play a role towards this immunosuppressive decrease in the tumor microenvironment. This is what they finally conclude, and that it's the antigen presenting CEF that increased and this myofibroblast CEF that decreased, that leads to this oncolytic herpes simplex virus to act on the tumor, as this will help in early detection of this tumor by the immune system. So that's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.